The forgotten inventor of antibiotics. 16 years before Alexander Fleming discovered the bacteria-inhibiting properties of mold, a French scientist had described a similar discovery in his doctoral thesis. In 1897, a young French medical student named Ernest Duchesne submitted a doctoral dissertation titled Contribution to the Study of Vital Competition in Microorganisms, Antagonism between Molds and Bacteria. In this thesis, Duchesne proposed a revolutionary idea that bacteria and molds are constantly engaged in a survival struggle, and humans could harness this antagonism to treat diseases, according to Amusing Planet. Although the medicinal properties of molds and plants in treating infectious diseases had been known since ancient times, Duchesne was the first to experimentally demonstrate that certain molds could destroy disease-causing bacteria such as Salmonella typhi, which causes typhoid fever, and Escherichia coli in laboratory settings by injecting them into guinea pigs. What Duchesne discovered was natural penicillin, a breakthrough often associated with Scottish physician Alexander Fleming. Duchesne's work was almost forgotten until it was rediscovered more than 50 years later, in 1949, 4 years after Fleming was awarded the Nobel Prize. Ernest Duchesne was born in Paris in 1874, the son of a chemical engineer who owned a tanning factory. After finishing high school, he enrolled at the Lyon Military Medical School, École du Service de Santé Militaire, in 1894. 2 years later, Duchesne began researching under the guidance of Gabriel Roux, a professor of microbiology and the director of the Provincial Sanitation Office in Lyon. Roux observed an interesting phenomenon. Although mold spores were abundant in the air, they were absent from tap water and fountains, even though they could grow in distilled water. This led Roux to suspect that some microorganisms in water might inhibit mold growth. He suggested Duchesne explore this idea as the foundation for his thesis. This observation became the starting point for Duchesne's core research on competition between microorganisms, eventually leading to his discovery of penicillin. Duchesne conducted a series of experiments in which he cultured Penicillium glaucum in meat broth and then introduced small amounts of Salmonella typhi and Escherichia coli into the mold population. Each time, the bacterial spores died. Duchesne concluded that in the struggle for survival, bacteria were initially more dominant. However, Duchesne speculated that before penicillium perished, it could weaken bacteria, thus reducing their virulence and disease-causing properties. To test this hypothesis, Duchesne injected guinea pigs with a solution containing equal amounts of Penicillium glaucum and E. coli. At first, the animals became seriously ill, but they recovered quickly. 2 days after the first injection, Duchesne administered another dose. The guinea pigs showed no signs of illness, indicating that they had developed resistance to E. coli. A similar immune response occurred when guinea pigs were injected with a mixture of S. typhi and P. glaucum. Duchesne observed that mold, Penicillium glaucum, when introduced into animals alongside certain bacteria, such as S. typhi and E. coli, could significantly reduce their virulence. While Duchesne could not isolate the antibiotic substance produced by Penicillium glaucum, he correctly concluded that molds could be used to treat diseases. Duchesne's thesis earned him his doctorate, but his ideas did not attract attention from the medical community. By the end of 1898, Duchesne was appointed as a doctor in the 2nd Cavalry Regiment, stationed in Senlis. He married in 1901, but his wife died 2 years later from tuberculosis. He himself fell ill in 1904 and was discharged from the army in 1907. Duchesne spent his final years in various sanatoriums in southern France and Switzerland before passing away in 1912 at the age of 37. In 1928, 16 years after Duchesne's death, Alexander Fleming made a similar discovery when his petri dish of Staphylococcus aureus was accidentally contaminated with penicillium mold. Like Duchesne, Fleming observed that the mold secreted a compound that inhibited bacterial growth. Despite its significance, Fleming's 1929 paper, published in the British journal Experimental Pathology, failed to attract much attention, much like Duchesne's thesis years earlier. Fleming himself was unsure of the practical medical applications of his discovery. He focused more on its potential use in isolating bacteria rather than treating infectious diseases. His colleagues in chemistry attempted to isolate the active penicillin compound but were unsuccessful, leading Fleming to discontinue further research. A decade later, in the late 1930s, British biochemist Ernest Boris Chain rediscovered Fleming's overlooked 1929 paper. Recognizing its potential, Chain suggested to Australian scientist Howard Florey to investigate the antibacterial properties secreted by microorganisms. Florey assembled a team of biologists and biochemists at the University of Oxford. Their collaboration eventually led to the successful isolation and mass production of penicillin, turning it into a life-saving antimicrobial. This breakthrough came just in time for widespread use during World War II, transforming modern medicine. After the therapeutic properties of penicillin became widely known, Alexander Fleming received widespread recognition, overshadowing Howard Florey. In 1945, Fleming, along with Florey and Ernst Boris Chain, was awarded the Nobel Prize in Medicine for the discovery of penicillin and its curative effect in various infectious diseases. Although the prize was shared equally among the three, Fleming continued to be the public face of penicillin, while Florey and Chain's critical roles in making penicillin a viable treatment were often overlooked.